are at the beginning of the food chain. And like this, our life is fully supported by photosynthesis. It's an important, unique process. And also, photosynthesis is important for bioeconomy, for blue biorefineries, because photosynthetic microbes can offer sustainable production platform. Cyanobacteria can run photosynthesis, absorbing light, and for make inorganic compounds and biomass. And for producing this biomass, it needs only CO2, it needs water and some minerals, and of course sunlight. And advantage of this system is that it can take efficiently CO2. You can go up to 20-40% of CO2. They can still be happy and produce even more biomass. They can use industrial flue gas, and you don't need also to provide them with the clean water. You can give them industrial wastewater. And produced biomass is called 3G biomass, third generation biomass. And this biomass can be used in blue biorefineries to make biofuels, chemicals, food, and feed. Cyanobacteria also has advantage because they are growing very fast. Some cyanobacteria can have few hours doubling time, which is quite big advantage. So photosynthesis is the starting point for bioeconomy, biorefineries also. Photosynthesis, if we talk about the photosynthesis, we can describe it as two sets of the reactions. Here is the simple reaction of photosynthesis. And in these two sets of the reactions, in the first part is called light reactions. These light reactions occurs on thalloid membrane. Cells, photosynthetic organisms, they capture the light energy and they use this light energy to split water to oxygen. At the same time, they use this light energy to produce ATP and NADPH. This ATP and NADPH stores energy from light in a chemical compound. And this ATP and NADPH can be used by dark reactions to fix CO2 into organic matter. And during this process, low energy oxidized carbon in CO2 is fixed to high energy reduced carbon. And this molecule stores light energy in the structure of the chemical compound. The process, these dark reactions, also called carbon reactions, and it's a little bit misleading because actually these dark reactions occurs in light, but they are not light dependent, and that's why they are called dark reactions. As a summary, the end product of the light reactions are ATP and NADPH, which further is used to fix the CO2 to sugars. Photosynthetic light reactions, they take place in thalloid membrane. This is the cyanobacteria. And if you look on the microscope, you can see that on the microscope, they are gram-negative bacteria and they have a typical cell envelope. So they have an outer membrane, they have a plasma membrane, and also inside of the cells, in the cytosol, they have a thalloid membrane. And this thalloid membrane is layered inside of the cells, and they are also they are interacting with plasma membrane. And inside of this thalloid membrane, you can see also lumen. And if you focus to thalloid membrane, you will see this kind of structure. All four big photosynthetic protein complexes, they are embedded to thalloid membrane. These are photosystem 2, cytochrome BF complex, photosystem 1, and ATPase. Light captured usually by big, huge antenna complex, which is called phycobilisomes in cyanobacteria. These phycobilisomes are attached to thalloid membrane. And I would like to mention that there are some exceptions. For example, Glebobacter is the only cyanobacterium which does not have a thalloid membrane. And during this photosynthetic process, there is a PMF, protomotive force, generation which runs the ATP synthase. I will come back to this. So phycobilisomes are peripheral photosynthetic antenna complexes. They attach to thylakoid on cytosol side. And there is a difference between algae, plant, and cyanobacteria because in case of the green algae and plants, they harvest light energy through the light harvesting complex, which is embedded to thylakoid membrane. In this case, it is peripheral, but in those cases, it is embedded to thylakoid membrane. And phycobilisomes covalently bind phycobilins. There are different phycobilins, phycoerythrin, phycocyanin, allophycocyanin. You can see here that they catch the photons, they transfer the light energy through the phycobilisome rods, and eventually they end up in the core of the phycobilisomes, which is the allophycocyanin. They have different maximum for absorbance. Phycoerythrin maximum absorbance is the 570 nanometer, for allophycocyanin is the 650 nanometer, and eventually the light comes and captured by chlorophyll A of photosystem 2, primary donor. It's called P680. This is the chlorophyll pair. And when P680 captures the light, it becomes excited, and then it can eject the electrons to the next acceptor. Like this, light charge separation occurs and photochemistry starts to work. If we talk about photosynthetic electron transport, it's much easier. We clarify it, we describe it by Z scheme. Here you can see simplified Z scheme. The electron transfer starts from photosystem 2 according to Z scheme. The light punctures the P680, the primary donor of photosystem 2. After excitation, it ejects the electron to the acceptor, and it becomes P680 plus. P680 plus is the strongest biological oxidizing agent. It has a very high redox potential, and it is so strong biological oxidizing agent that it is able to split water, to take out from the water oxygen and protons. At the same time, when light is shining, also P700 becomes excited, and P700 is the primary donor of the photosystem 1. And again, electrons ejected to the next molecule, into the next acceptor molecule. And P700 excited is a very strong reductant. It is also so strong reductant that it is able to reduce NADP plus to NADPH. And between these two photosystems, you can see cytochrome BF complex, which mediates the electron transfer between photosystem 2 and photosystem 1. I come very shortly to photosystem 2 because it's a really unique system. It is only protein pigment complex which is able to oxidize water. Photosystem 2 in cyanobacteria is usually as a dimer, but you can see here a monomer of that photosystem 2. And each monomer has about 20 protein subunits and different cofactors for electron transport. And D1 and D2, they are the heart of the photosystem 2. Almost all electron transport cofactors are on these two proteins. When P680 becomes excited and charge separation occurs, pheophytin gets the electrons, and electrons from pheophytin goes to QA, which is the plastikinone molecule, and then from QA to QB, which is another plastikinone molecule, and sits in QB site in the pocket. And after two charge separation, QB receives 2 electrons and takes 2 protons from cytosol, becomes double protonated, reduced, and migrates to plastikinone pool. But P680 plus is very dangerous molecule because it needs to be reduced immediately. And it can take electrons from water oxidation complex and split water. This water oxidation occurs in manganese-calcium cluster. This cluster consists on 4 manganese ion and calcium ion. And you can see that this cluster can remove, this is the different flashes. While you give the different flashes, it can remove 4 electrons and 4 protons. It can split water and to form the oxygen. So like this, these electrons go back and reduce P680. There is the extrinsic proteins also, which shield the manganese cluster because it needs strong protection. We come back to thalloid membrane. So electrons now passed to QB. QB double protonated and migrated to plastikinone pool. And from plastikinone pool, it can bind to cytochrome BF complex. Like this, cytochrome BF complex can take 2 protons and pump to lumen and give 1 electron back via cytochrome B6 to plastikinone, the another electron via cytochrome F to plastocyanin or cytochrome C6. These plastocyanin and cytochrome C6 are small mobile electron carrier proteins. They are working between cytochrome BF and photosystem 1 to pass the electrons from cytochrome BF to photosystem 1. And electrons from plastocyanin can reduce primary donor of the photosystem 1 P700 back. Electrons from photosystem 1 eventually ends up to reduce ferredoxin FNR and reduce NADP to NADPH. So if we look to whole photosynthetic linear electron transport, the water-based electrons or electrons coming from the water splitting at the end up to reduce NADPH, which is reducing power for carbon fixation and different cell metabolism. During this process, if you pay attention, there is a proton pumping from cytochrome B6F, and also protons are in lumen coming from water splitting. So there is a delta pH difference across the membrane. At the same time, there is also charge separation in both photosystems, and there is also membrane potential. We call it delta psi across the membrane. And these 2 components, delta pH and membrane potential, create proton motive force across the membrane. And this proton motive force runs ATP to produce from ADP ATP. ATP and NADPH later on used for cell metabolism. Okay, so we know what happens in thalloid membrane, and it looks pretty simple, but we have to consider that cells are always experiencing different environmental conditions. There is always fluctuations of the CO2 amount, fluctuation in nutrient availability, fluctuations in light intensity, quality, temperature fluctuation. These all should, this or other way, affect the photosynthetic apparatus. So what happens if photosynthetic electron transport chain exposed to different environmental conditions? Light reactions, we already discussed, and these light reactions coupled with CO2 fixations. For example, if there is a low temperature, for some reason CO2 fixation is not very efficient, CO2 fixation enzymes working slowly, what happens then? Then, of course, there is an accumulation of the electrons in light reactions in photosynthetic electron transport chain in thalloid membrane. Light is absorbed, there are a lot of electrons, but they cannot be used for CO2 fixation or downstream processes. There is an accumulation of electrons, and that electrons should go somewhere. They find oxygen because this is oxygenic photosynthesis. There is a lot of oxygen available. So those electrons can interact with the oxygen and produce reactive oxygen species. It can be singlet oxygen, it can be superoxide radical, and so on. These reactive oxygen species, actually, they are important as signaling molecules. However, if there is a massive amount of the reactive oxygen species, they attack photosynthesis, they attack photosynthetic apparatus and they damage photosynthetic pigment protein complexes. And that's why the cells, they have evolved many different protection mechanisms, many different alternative electron transport routes to be able to overcome stress conditions and to keep photosynthetic apparatus functional. So this is what I have shown you previously, the simple electron transport in thylakoid membrane, including 4 big protein complexes and 2 photosystem 2 and photosystem 1 is pigment protein complexes. But there are many players which are involved in fine-tuning of photosynthetic electron transport and in regulation of photosynthetic electron transport to be able to avoid stress conditions and excess pressure to photosynthesis. In our lab, we are studying many of these auxiliary electron transport routes, and I will show you some examples to understand how the process goes with the protection. If we try to somehow summarize these protective mechanisms, I will focus mostly to electron sinks, and I will mention also cyclic electron transport. Cyanobacteria, also another photosynthetic organisms, they have a strong electron sinks. That electron sinks are important when electron input and outlets are not well balanced. So one of those electron outlets are fluorodiiron proteins. You can see them there. Fluorodiiron proteins are a large family of enzymes. They can be found in archaea, in bacteria, in some eukaryotic protozoan pathogens. Mainly they function in oxygen and NO detoxification. Anaerobic bacteria, when suddenly exposed to oxygen, they can use this reaction to eliminate the oxygen. And they have a 2 conservative domain, you can see here. And another important thing is that fluorodiiron proteins, they are around 60 kilodalton proteins, but they are not working as monomers, because the distance between reaction centers, FMN and ferrum here, is quite long for electron transfer. And that's why they make heterodimer or heterotetramer, homodimer or homotetramer, let's say homooligomer, and they make a head-to-tail arrangement where one reaction center, ferrum faces FMN, and then this distance between them is quite close, and electron transport can occur very quickly. In general, in photoautotrophs, fluorodiiron proteins are quite much found in almost in all photoautotrophs. And in photosynthetic oxygenic photoautotrophs, they have specific domain which can bind reduced ferredoxin or NADPH from photosynthetic reactions, and it can give that electrons to oxygen and become NADP plus or ferredoxin. In green algae, in cyanobacteria, in mosses, you can find fluorodiiron proteins, but during evolution, they disappear from angiosperms for some reason, from Arabidopsis, for example, from tobacco. You cannot find fluorodiirons there. And in Synephacystis, because this is some kind of model unicellular cyanobacteria, that's why I mentioned Synephacystis, it has 4 fluorodiiron proteins, and they make somehow heterodimers. We know that fluorodiiron 4 and 2, they make heterodimer and they are in photoprotection. And FLV1 and 3 also make a heterooligomer. They function also in oxygen photoreduction. And important to mention that on the ambient level, we have a lot of fluorodiiron proteins. You can see in wild type, this is the amount of the fluorodiiron 3, this is the fluorodiiron 4. But if you go high CO2 level, like 1% or 3% CO2 level, when you grow the cells under high CO2 conditions, then you can see strongly down-regulated fluorodiiron 3. If you compare to this one, it's much less. And actually, fluoroiron 2 and 4, they are expressed as an operon. They are very low on protein level. It is hardly detectable. And I would like to mention that fluoroiron proteins brought to this field by Aaron Kaplan group already in 2003. And there they used high CO2 grown cells, and they clearly show that fluoroiron 1 and 3, they are involved in Mehler reaction. Mehler reaction is when the cells give electrons directly to oxygen. And after that, all these fluoroiron-based studies started, including our laboratory. And to be able to study fluoroiron proteins, we set up membrane inlet mass spectroscopy. If you think that fluoroiron proteins can reduce oxygen under light, to be able to differentiate, to see that function, we need isotope. Because cells, they split water, they produce oxygen. And if you check with the simple Clark type electrode, which is in every lab, what you see the oxygen uptake in the darkness. And when you put light, you see oxygen evolution. But this is a net oxygen evolution because there is certain oxygen is evolved by photosystem 2, but at the same time, in the darkness, there is some amount of the oxygen is consumed by terminal oxidase and fluoroiron proteins. You cannot differentiate it in Clark type electrode. You need isotope and you need membrane inlet mass spectroscopy. In this case, we add isotope to the medium. And when we see the oxygen evolution, this is the gross oxygen evolution coming from water. And we see also oxygen uptake because we can monitor oxygen 18 and we can see that there is an oxygen uptake in the light. And then we can subtract and find the net photosynthesis. Indeed, if you go with the mass spectroscopy, you can see that when you shine the light, wild type synchreticistic shows very strong oxygen uptake, which is missing in fluoroiron deletion mutant, which means that these mutants are responsible for light-induced oxygen uptake in synchronicity. Then we were thinking a lot that how we can find the phenotype in the mutant, because if a lot of electrons can run from water splitting to again make a water at the end of the photosystem 1, then this is the waste of the electrons. When we delete those waste of the electrons, we have to see better growth or something should happen to the cells because we delete also protection mechanism. After some thinking, we have decided to make a fluctuating light setup because in the water, in aquatic system, there is a lens effect of the waves. And this lens effect, when there is a wind or so on, it can produce really excessively strong and also dim light. You can see here in the figure also that there is very strong light or there is also dim light. And what we did, we used the 20 microeinstein background light in phytotron, and every 5 minutes, we used 500 microeinstein highlight pulses for 30 seconds. Every 5 minutes, 30-second highlight pulse. And in another setup, we made a little bit milder fluctuating light. Background light is 50 microeinstein, and still pulses 500 microeinstein. And when we use the severe fluctuating light condition, we see that the mutants which are lacking fluoroiron proteins, they are not able to grow at all. But if there is a mild condition, they can grow, but they grow much slow compared to wild type. So we made a conclusion that fluoroiron proteins 1 and 3 enable cyanobacterial growth and photosynthesis under fluctuating light conditions. And the main target during this fluctuating light is photosystem 1. You can see that at the third day of the exposure to fluctuating light, photosystem 2 protein, the core protein D1, is at the same level like at 0 day, But photosystem 1 protein is strongly down compared to the raw. And this is because when you put the highlight, the P700, which is the photosystem 1 primary donor, immediately becomes oxidized in wild type. But in the mutant, there is a lag phase. It takes several seconds until it starts to be oxidized. And this lag phase, because P700 cannot be oxidized, it creates reactive oxygen species and it damages the photosystem, and eventually the cell cannot survive under strong fluctuating light conditions. Coming back to high CO2 and low CO2 conditions, because I mentioned that these proteins are quite much abundant under low CO2 conditions, but under high CO2 conditions, we see on transcript level very less transcription, and also protein accumulation is very low. Again, back to the immunoblot. You can see that on the ambient layer level, we have flavodiiron 2, 4, and 3 quite high level compared to high CO2, 3% CO2. There is some FLV3, but it is relatively low. But we practically don't see flavodiiron 2 and very tiny amount of the flavodiiron 4. If we see such accumulation level of the flavodiiron proteins, then the first thing comes to mind that most probably under high CO2 condition, oxygen photoreduction will be very low. But when we did the membrane-late mass spectroscopy we performed under two different CO2 conditions, we see that this is not the case. So this dot line is the cells when we perform membrane-late mass spectroscopy with the cells grown under low CO2. Darkness, there is some dark respiration, and here we put a light, immediately strong light-induced oxygen uptake, and then it slowly decreases a little bit down. But if we grow the cells under high CO2 conditions, we have very strong oxygen uptake, the same level like a low CO2, but it relatively stays longer period, stable, which means that even on protein level, flavodiiron proteins are less in high CO2 grown cells, but still they are able to keep strong Mehler reaction under these conditions. And of course, we need the evidence that this oxygen uptake comes from the flavodiiron proteins. You can see here that if we delete flavodiiron proteins, there is no strong oxygen uptake. This one is the deletion mutant. So which means that the majority of the oxygen uptake under light comes from flavodiiron protein 1 and 3 under high CO2 conditions. We have done another experiment. I don't go to the details, but I come to the scheme to explain the current situation, which means that flavodiiron 1 and 3 make a heterooligomer and they function in oxygen reduction. At the same time, in the cells, you can find flavodiiron 3 homooligomers because we use the different mutants to prove it. And we do see photoprotection, but we don't see oxygen uptake, which means that these homooligomers are functioning in photoprotection, however, their exact role is still unknown. Then we go to air level CO2 condition. In the same immunoblot and the air level, we have huge amount of flavodiiron 3, 4, and 2 compared to high CO2. And in this case, we have flavodiiron 2 and 4, which we did not see on the high CO2 because they are not accumulated well in high CO2. But in low CO2, ambient level air, there is a flavodiiron proteins. And we do see that these flavodiiron proteins also contributes to oxygen photoreduction. And that's why I put them here. And we are also suggesting, and we are working on this to clarify that there is also different combination of the heterooligomers of the flavodiiron proteins 1 and 4. So to summarize all, what we can tell that under high CO2 conditions, flavodiiron 1 and 3 is mostly responsible for steady state oxygen uptake in cyanobacteria. and it can take about 20-60% of the electrons coming from water and give it back to water, make a water-water cycle. However, I would like to mention that we are talking about the transition from dark to light or from low light to high light. And in case of the air level CO2, there is two types of the heterooligomers. Flavodiiron 1 and 3 in this case is involved in transient and very strong electron sink, but there is also flavodiiron 2 and 4, which are also involved in oxygen photoreduction, but they are relatively slow and relatively less capacity for oxygen photoreduction. In any case, these flavodiiron proteins are the most strong electron sinks in cyanobacteria. The other electron sinks in cyanobacteria are respiratory terminal oxidases. Synechocystis has 3 sets of the terminal respiratory oxidases. These are cytochrome C oxidase, COX, cytochrome BD phenol oxidases, we call it in Finland SUT, and phenol oxidases or alternative respiratory terminal oxidase, it's called ARTO. This ARTO is mostly in plasma membrane, and so far it was not proven that it affects much to photosynthesis. That's why I don't mention it, but I will mention these 2 respiratory terminal oxidases. And we are facing the same problem. We know that these respiratory terminal oxidases function in respiration in dark. But do they function during the light also? And how strong they can be as a sink under the light? This is the problematic because, first of all, again, the same situation when there is oxygen produced by water splitting, by photosynthesis, and possible oxygen uptake by terminal oxidases in the light. So we have to differentiate. We can differentiate it again with the oxygen isotope. But there is another problem is related to flavodiiron proteins, because I mentioned that flavodiiron proteins are functioning in oxygen uptake, and they are very strong electron sinks. So they can easily mask the function of the respiratory terminal oxidases. For that one, we need different inhibitors and mutants to be able to localize terminal oxidases in which part of the thylakoid membrane they function. We use DBMIB. DBMIB is inhibitor of cytochrome BF complex. It affects at this level, which means that if you add this inhibitor, you don't see any more effect of the COX and flavodiiron proteins, but you will see what happens until cytochrome BF complex. And then another inhibitor is the HQNO, and this is not really specific for phenol oxidase, but it inhibits phenol oxidase, and it also has an effect to cytochrome BF. And then we always use also mutants because inhibitors have really many adverse effects. So it's better to validate the data with mutants also when it's possible. So let's look again photosynthetic electron transport. To be able to see possible light-induced oxygen uptake by phenol oxidase, we add the BMIB, we cut electron transport at cytochrome BF level. Before that, we looked at control wild type. This dark is the dark respiration. And then when we put light, we can see light-induced oxygen uptake. You see this light gray is the light-induced oxygen uptake. Dark respiration and light-induced oxygen uptake. This is the total oxygen uptake under the light. And when we cut the electron transport at the cytochrome BF complex level with the BMIB, we see some increase of dark respiration. There is some artifact also. We don't talk about this one now, but what we see is clearly light-induced oxygen uptake. You can see it here. So when you cut electron transport, there is a still outlet of the electrons to oxygen from this part. And what it could be phenol oxidase. And if we add the phenol oxidase inhibitor HQNO, we don't see light-induced oxygen uptake anymore. But again, I'm telling that since these inhibitors are not really so specific and they can affect whatever, it's always good to use the mutants also to validate that. So that's why we use the deletion mutant of the phenol oxidase. If you have a deletion mutant of phenol oxidase, and if you don't have any these inhibitors, you see strong light-induced oxygen uptake because electrons go to COX and electrons go to flavodiiron proteins. But if we cut here the cytochrome BF and this is lacking in the mutant, then there is no light-induced oxygen uptake similarly to this inhibitor study, which once more shows that phenol oxidase is functioning at the level of the plastikinone pool and it takes electrons from photosystem 2. And this also shows that flavodiiron protein 2 and 4 are not working at plastikinone pool level. Because in previous our studies, we suggested that they might take an electron from photosystem 2 and protect photosystem 2. But if we rely to these experiments, there is no electron outlet to oxygen at plastikinone pool level, at least for oxygen. So it can be that flavodiiron protein 2 and 4 working there, but they have different function. We don't know yet. Here is the summary of this part, and if we go back, we can tell that flavodiiron proteins have very high capacity, up to 20-60% of the electrons coming from water to give back to water and to work as an electron valve, as a protection mechanism. And we can tell that respiratory terminal oxidases, these two, they are also working in thylakoid membrane. They also contribute to protection, but we have to make clear that these SUD and COX, they do not have as much as capacity like flavodiiron proteins. They cannot handle that amount of the electrons like flavodiiron proteins. However, it's clear that these respiratory terminal oxidases continue to work under the light, but on slower time range and in limited scale. Shortly about cyclic electron transport. Cyclic electron transport around photosystem 1 is important because photosynthesis during the linear electron transport produces NADPH and ATP, but that ATP is not enough to run carbon reactions and for cell metabolism. That's why photosynthesis needs extra ATP for cell metabolism. And like this, the electrons coming from photosystem 1 is cycled back to plastikinone pool or cytochrome BF, and this cycling creates PMF, proton motive force, which creates extra ATP. However, it does not create NADPH. So an ATP NADPH ratio is also balanced by cyclic electron transport. There are several routes for cyclic electron transport. PGR5-like protein is the one route. So PGR5 is playing a role in cyclic electron transport, or to be correct, in creating, generating proton motive force in Arabidopsis, in plants and in algae. And Cyanobacteria has also small protein which is called PGR5-like protein because it has relatively less homology. And already many years before, Hans Matisse group showed that PGR5 might have a similar cyclic electron flow function in Cyanobacteria. After that, for several tens years, more than 15 years, it was silenced, and recently come several, again, new publications where they argue that indeed in PGR5 might be involved in cyclic electron flow. It can take electrons and it can be somehow involved in this electron flow, but this road still needs more clarification. There's an NDH1 complex in Cyanobacteria. You can see it here. And this complex has huge contribution to cell metabolism and to photosynthesis. This NDH1 complex is homologous to mitochondrial bacterial complex 1. And recently it has been confirmed, it has been shown that it can take electrons from ferredoxin and give it back to placental pool, and like this to stimulate also cyclic electron flow around photosystem 1. Cyanobacteria has different NDH1 complexes, and depending on its subunit composition, NDH1 can function in cyclic electron flow, and it can function in respiratory electron flow, and it can function also in CO2 uptake. So this is the NDH1L complex, large complex. They are mostly in cyclic electron flow around photosystem 1, and also in respiration. And these are NDH1MS complex involved CO2 uptake and also to some extent cyclic electron flow. And if we come to our scheme, you can see that NDH1 complex takes electron from ferredoxin, and flavodiiron proteins also takes from ferredoxin. And the work with applying different mutants and studying this function of different mutants, we show that there is some kind of coordinated action between flavodiiron proteins and NDH1 complex. And we are also interested that what would happen if we delete both, for example, flavodiiron 1 and 3 and NDH1L complex at the same time. And what we see that this is the NDH1 D1, D2 mutant, which is the deficient in NDH1L complex. This is the flavodiiron protein 1 and 3 mutants, and this is the triple mutants. So we see that if these triple mutants cannot survive conditions, when you make a transition from high CO2, moderate light, like a 50 micron to air level CO2 and high light, like a 220 micron. This combination, the deletion of the both flavodiiron proteins and NDH1L complex, is lethal for the cells. They cannot accumulate to these conditions. This gives us also possibility to tell that there is a functional redundancy between flavodiiron proteins and NDH1 complex. There is a small protein also, I quickly mention about it. This is the bidirectional ferronickel hydrogenase. It's also called HOX hydrogenase. And this HOX hydrogenase, it's a heteropentameric enzyme. It has a catalytic core, say HOX-H and HOX-Y, and diaphragm moiety. There is not much known about the function of this protein. What we know that if cells are under anaerobic conditions, then when you expose the cells which are under dark anaerobic condition to light, then you can see tiny amount of the hydrogen production in synthesistis, which is coming from this HOX enzyme. And immediately when oxygen occurs under the light, then hydrogen production goes down, the HOX becomes inactive. And if we use the different mutants, like a flavodiiron mutants or like a triple mutants, flavodiiron and NDH1 complex, you can see that you can get more hydrogen production via HOX, which means that if you delete these roads, then you can funnel more electrons to HOX. And also HOX has different additional role besides the hydrogen metabolism. It needs further clarification. So I would call that there is a lot of research around this protein still to be done. So putting everything together, what we can tell? We can tell that photosynthesis runs linear electron flow, takes light energy, split water, and converts this light energy into chemical energy of the NADPH and produce the ATP. In the second round, when cyclic electron flow happens, the red line, the photosystem 1 takes the light energy, runs the system, reduces ferredoxin, and goes back again, cycles around photosystem 1. Like this, the cells produce ATP via ATPase, but they do not produce an ADPH. And this is important under certain stress conditions to be able to get more ATP. And then cells also can use the water-water cycle when water is splitted at photosystem 2. Electrons run through all these cofactors, and at the end, again, it ends up in water via flavodiiron proteins or via terminal oxidases. And in this case, water-water cycle serves to extra ATP synthesis, and also it functions as a valve temporarily. Why it is important to know all this regulation of photosynthesis? It's important because we can funnel more photosynthetic electrons to the desired end products to make a more efficient cell factories. I would like to mention that theoretical light to product conversion efficiency in photosynthesis is pretty high. It's around 10-13%. However, in current situation, due to all these different regulation processes, protective processes, this light to product conversion efficiency is under 1%. If we would be able to increase this light to product conversion efficiency, we will be able to get more photosynthetic electrons to chemicals and to make cyanobacterial production system more sustainable. I would like to thank to my group, Photomicrobes group. We have several new members just started in September, but because of the COVID, we don't have the possibility to take a new photo. And here is our funding agencies. We have a very extended network of the different with different European photosynthetic groups. I did not list them here because I give very general lecture here, but I'm very grateful to all of them. And at the end, I would like to mention you about our Nordic Center of Excellence, which is coordinated by Turku University. I'm chairing this Center of Excellence. And here we have 10 different partners from 3 different universities around Nordic countries. And our aim is to make a algae cyanobacteria-based sustainable production system. And as you can see, one of the work packages is improvement of photosynthesis. So we do hope that we can improve photosynthesis and to have a sustainable bioeconomy for future. Thank you for your attention.